Hey, my name's Vicky Brown. I'm one of the biology teachers at St John Rigby. So when you come to us to start your A-level biology course, I might be one of your teachers. There's Sue and Kate who works alongside me. We are hopefully producing some videos and things for you to help you bridge from your GCSE studies to your A-level studies. Obviously we're home, as you can see, not at the college at the moment. So I apologise for the quality of the video later on, but we're doing our best to make sure we bring you some lessons in this time. So this is how it's going to go. I'm going to show you my PowerPoint. We're going to do lots of activities so that you can stop this video, have a go, and then join me back to check your answers. And by doing this, we'll recap and check all of your cells knowledge in this video. So there are 4 videos that we've put together for you. This one's to get you ready for the cells topic. Okay, so let's get started. Okay, so as you can see, hopefully you can see the PowerPoint in front of you. You've got access to this PowerPoint as well to have a play with and review again closer to the time starting college. Okay, so what I'm going to ask you to do is to pause at intervals and have a go at the activities. So as you can see on the screen, we've got the starter activity. Okay, so if you could pause here and have a go and then rejoin me, we'll go through the answers together. Okay, so you should have joined me back now, and we're going to have a look at the answers. So the first question was to match up these cell types with the diagrams, and we had plant cell, algae cell, animal cell and bacterial cell. So looking for the plant cell first of all, so I'm hoping you've picked D there for that one, and that's because the plant cell has got the permanent vacuole. You can see the chloroplast. You can also see a cell wall. Okay, so they're all the clues. That was your plant cell. Your algal cell, I think you'll probably have left till last. Okay, so that might be the one that you did at the end when you found your others, because that will be the one you're less familiar with. Okay, so that's A, and we'll come back to that. The animal cell, of course, is B. So just the cell membrane visible and the nucleus. And finally then, C is our bacterial cell. So the bacterial cell we can tell because it has no nucleus and its DNA is a circular molecule free in the cytoplasm. So this is a loop of DNA free in the cytoplasm. It's also got a cell wall. Okay, so there's your correct answer for those. Hopefully we've got those so far. If not, don't worry, because we'll be recapping some of those key features as we go through using some other activities too. Okay, so question 2, identify which of these cells are prokaryotic and explain why. Okay, so for this one, I'm hoping you've pulled out C. For the reason I pointed out earlier, the DNA, the genetic material, is free in the cytoplasm, so it's not in a nucleus like the other cells. So A, B and D are all eukaryotic cells with a true nucleus, whereas prokaryotes do not have a nucleus, and that's the evidence we're looking for in that cell. And then finally for 3, give the carbohydrate found in the cell wall of cell A and D, but not in C. So we should know that our plant cell walls and our algae cell walls contain cellulose. The prokaryotic cell has a cell wall, but it is not made of cellulose. Okay, so that's a structural difference. Right, so big picture. In your GCSE studies, what you will have been understanding is that cells are the basic unit of all forms of life. Okay, but the big picture to that is that some forms of life on the planet are made up from just single cells. So things like bacteria, so that's our prokaryotic example, but there are also things like yeast and algae that are eukaryotes that are single cells too. Then you would have been thinking in your GCSE studies about the fact that some organisms are more complicated than that. They're multicellular organisms, And so they're made up of many cells that are arranged into tissues and organs and body systems. So once we've thought about single cellular organisms and multicellular organisms, we've also in that cells topic at GCSE looked at cells of organisms from different types, like animal, plant cells, bacterial cells, and we found that they have some structural differences, okay, which allows us to identify them like we did in that first task. We looked at these structural differences in these different cell types of different types of organisms to be able to identify them. Okay, so that's what you've been busy doing. Okay, I'm going to recap some of that. Just in case you're wondering, in the pictures over there, we've got a single cellular algal cell under the microscope. We've got lots of single cellular bacterial cells. And we've also got some human cells arranged into tissues and organs and an organ system. Okay, so we're going to start with the animal cell, and I'd like you to pause here and draw and label this cell using the labels at the bottom. Okay, so you should be back with us. We're going to go through those labels. Okay, so let's see if you've correctly labelled your cell. So the first one we're looking for was the nucleus. So we want this structure here labelled as the nucleus. The next one we want the cytoplasm. Okay, so this is the space between the subcellular structures. So it doesn't matter where you've labelled that up as long as it's the space between these other structures. Then you want the cell membrane, which is the boundary, outer boundary to the animal cell. And the mitochondria. And finally the ribosomes. Okay, so we will need to know some of these structures in terms of, we need to know their functions as well. So a bit of a recap. Can you remember the function of each of these labelled structures? Okay, so we're going to go through each one. So think about the cell membrane. What's its function? Well, we would say the function is to control the movement of molecules into and out of this animal cell. What about the cytoplasm? What's the role of the cytoplasm in the cell? Well, it's the site of chemical reactions within the cell. Right. What about the ribosomes? They have a very important function. I know you met them in your protein synthesis topic. So they are the site in the cell where proteins are synthesized. The nucleus next. The nucleus, you can say, controls the activities of the rest of the cell, specifically because it contains the genetic information, which codes for all the proteins the cell can make. And the mitochondria. Well, that is the site of ATP production. Okay, so ATP is an energy storage molecule. So when we describe what happens in the mitochondria, it's where respiration takes place, where the energy trapped inside sugar molecules is converted into energy trapped within ATP, which can then be used, that energy, to power the chemical reactions in the cell and activities like active transport. Okay, moving on then to our next cell. Can I ask you to stop the video, draw and label using the labels, and this time can you challenge yourself to come up with the functions too? Okay, so you should be back with me. We're going to go through the labels first of all. So label number 1 should be our nucleus. Number 2, cytoplasm. Again, any space between the structures within the cell is fine. Cell membrane. Now this, you have to be very careful to make sure your arrow touches the inner boundary. Okay, that's your cell membrane. Mitochondria, up in this top corner over here, also some down here. Ribosomes. We've got 2 sort of groups of ribosomes in this cell. chloroplast, so a new structure in our plant cell, a permanent vacuole filled with cell sap, and the very outer boundary, the cellulose cell wall. So the nucleus, cytoplasm, ribosomes, cell membrane and mitochondria have exactly the same role in the plant cell as they do in the animal cell. So we just need to think about the roles of our new ones. Okay, but I'll do a quick recap through the others. So cell membrane, we said, controlled the movement of molecules into and out of the cell. Ribosomes were the site of protein synthesis. Cytoplasm was the site of chemical reactions in the cell. The nucleus controlled the activities of the cell as it contains the genetic information. And the mitochondria produced ATP, which is the energy source for all the activities in the cell, including active transport. So what are these three new ones? So the permanent vacuole filled with cell sap. Okay, so this really is about maintaining the turgor pressure within the cell. So it fills up with cell sap, and that gives a sort of pressure exerted on the rest of the cell, and that's what keeps plant cells turgid, rigid in structure. The chloroplast. Now this is a really important function, okay, and this is the site of photosynthesis. So this is converting light energy into chemical energy in the plant. Process photosynthesis. And finally then, the cellulose cell wall. Well, the cellulose cell wall provides mechanical strength to the plant cell, so it keeps its shape, its structure, and it makes it a strong cell. So it keeps its shape and gives it mechanical strength. That's really important because when water enters by osmosis, plant cells do not burst like animal cells would if excessive water entered them by osmosis. Okay, so can you now have a go at this? So I'd like you to compare and contrast the structure of a plant and animal cells. So we're looking for, in a compare and contrast, similarities and differences. Okay, so have a pause, copy it down, fill in the gaps, and then compare to my answer. Okay, so you should be back with me now. So the first one actually is a similarity. So both plant and animal cells contain the mitochondria. Okay, and we said that was for the process of respiration, which is the process by which we make ATP in the cell. The second comparative point is a different one. Animals cannot photosynthesize because they do not contain chloroplast, whereas generally plant cells do contain chloroplast for photosynthesis. Okay, I say generally because there are some plant cell types, as we will recap in a minute, that don't need chloroplast because they are not photosynthesizing cells. Okay, another similarity, both contain ribosomes, the site of protein synthesis. But then we've got two differences at the end. Animal cells do not contain large central vacuoles, whereas plant cells do. And animals do not have a cell wall, whereas plant cells do, and it contains cellulose. Okay, we're going to move on now to the prokaryotic cell. Again, can you draw this diagram and label it? And where you can, recall the functions of any of the structures. Okay, so having paused, you should be back now with a label diagram. Let's have a look at what you should have. Okay, so plasmids, first of all, are the small circular DNA molecules free in the cytoplasm of the bacterial cell. Cell membrane, again, is the inner boundary. Cell wall, the outer boundary. This time it doesn't contain the cellulose. The cytoplasm, again, is the space between structures. That's where we're going to label that. And then we have got at the centre a large free circular DNA molecule. So the reason this is different to our plasmids is it's a much bigger loop of DNA. And then finally, there are ribosomes, but they are much smaller than the ribosomes found in eukaryotic cells. So bacteria are only prokaryotic cells. So when you look at things that are common to other cell types, like the cell membrane, it still has the same function. So it controls the movement of molecules in and out of the cell. The ribosomes are still the site of protein synthesis. The cytoplasm is still the site of chemical reactions. The cell wall has a similar function to that of plants, in that for a prokaryote it gives the prokaryotic cell mechanical strength. So water can move in by osmosis and it can become really full with water but won't burst because of the cell wall resisting the osmotic pressure. So where have we got some differences? Well, we have no nucleus. Okay, so the DNA, which codes still for the proteins that the cell makes, is free in the cytoplasm. And now we've got these additional bits of DNA, and I know from your genetic engineering topic in your GCSE, you know that this circular DNA also codes for proteins, but the proteins it codes for include those that give this bacteria the characteristic of antibiotic resistance, and that those can be transferred between organisms. So for bacteria, they can actually swap those plasmids, but there'll be more of that in your A-level studies. In terms of function, the plasmids have accessory genes. So they are genes for proteins that are advantageous but are not essential to the life of a bacteria, whereas the essential day-to-day proteins are coded for on this free large circular DNA. Okay. So now, can you describe how a prokaryotic cell differs from eukaryotic cells like animal, plant and fungal cells or fungi cells? So stop and have a go and then join me back. Okay, now I have to apologise for my handwriting on the screen for this one. I tried to use my stylus. Okay, so we'll just pretend that we're reviewing a piece of work from a student with rather terrible handwriting. So let's look to their first comparative point. They've said prokaryotic cells have no nucleus, eukaryotic cells have a nucleus. So we would give them the credit for that. They've also said prokaryotic cells that their DNA is circular and free. Whereas in eukaryotic cells, they've said it's linear, so that's like the opposite of circular, so a long straight line. So as you did your double helix and you looked at your double helix, that long straight double helix is not joined back to itself in a loop like it is in the prokaryotic cell. And that's found not free, but found in the nucleus. The third mark point, the prokaryotic cell does not contain mitochondria, whereas all eukaryotic cells contain mitochondria. You could expand here and talk about plant cells containing chloroplasts and prokaryotic cells not containing chloroplasts, but we're looking for things we could say quite commonly for all eukaryotes. In both, they have cytoplasm. In both, they have cell membranes. So these are not differences, so they wouldn't get any credit for those. Okay. So have a think about how you structure a question when you're asked to describe how they differ. Okay, so it wouldn't be for 5 mark points, it would possibly be for 3, and it's just those top ones that we would write. On the new GCSE and the new A-level courses, the exam board has started to use a new command within the exam, and the new command is compare and contrast. So next, I'd like us to have a go at comparing and contrasting a bacterial cell with an animal cell. for 5 marks. Okay, so if you stop and have a go on paper yourself first. Okay, so let's have a look now at what a good one looks like. Okay, so this is a waggle. So at GCSE, in your exam, if you're asked that question to get a good solid 5-mark answer, this is the kind of structure we'd be looking for. Okay, so first thing I want you to do is compare and contrast your answer with this one. I'm going to read it through so you can in real time first of all. So bacterial cells are prokaryotic cells and human cells are eukaryotic cells. Bacterial cells are much smaller than animal cells. Bacterial cells and animal cells have cytoplasm, ribosomes, although bacterial ribosomes are smaller, and a cell membrane. However, a bacterial cell has a cell wall, which an animal does not. Within a bacterial cell, the genetic material is not enclosed in a nucleus. It is a single DNA loop, and there may be one or more small rings of DNA called plasmids. In an animal cell, the DNA is linear and enclosed in a nucleus. Animal cells also have mitochondria, which bacteria do not. So just do a bit of a comparison in terms of your answer, structure, the detail. Now they would have got 5 marks, okay? So I'm going to show you it marked now, okay? So the first line they wouldn't have got any mark points for because I think they were just getting going. Bacterial cells are prokaryotes and human cells are eukaryotic cells. It's not really answering the question. Well, then they do start to have a comparison, and there are both similarities and differences, which is what we need in the compare and contrast. On the new specifications at both GCSE and A-level, this is much more common, and that's where students sometimes fall down because they're very good at remembering differences, but not always very good at outlining similarities. So this student has done well because they've been able to say bacterial cells are smaller than animal cells. That was a valid mark point. That they've both got cytoplasm, which again was a valid mark point, and a cell membrane, another valid mark point. The ribosomes was down as a difference, okay? So the idea that they are smaller ribosomes in bacteria and larger ribosomes in animal cells. And then there is mark points as well for the DNA in the bacteria being free and circular, whereas in an animal cell it was linear and enclosed in the nucleus. And then there was one more mark point at the bottom for another difference. Animal cells also have mitochondria and bacteria. Bacterial, it says there, should really say bacteria do not. Okay, so the student has done enough for 5 marks there at GCSE in terms of their detail level. It's not without mistakes, but generally, you know, as in grammatical mistakes really, but the biology is sound enough for 5. Okay, so how did you do in comparison? Did you have similarities, like both have cytoplasm, both have ribosomes, both have cell membrane? Did you have differences? Bacteria have a cell wall. Bacteria DNA is circular and free in the cytoplasm. Bacteria do not have mitochondria, but animal cells do. Okay, so moving on to a quick recap. Okay, so what have we reviewed from your GCSE studies? Well, we've looked at animal and plant cells, eukaryotic cells, and we've reminded ourselves that they have cell membranes, cytoplasm, genetic material enclosed in the nucleus, and mitochondria. We've also remembered that plant cells have chloroplasts, a permanent vacuole, and a cellulose cell wall. We've also recapped that bacterial cells are much smaller in comparison. They have cytoplasm and a cell membrane surrounded by a cell wall, but this time it doesn't contain cellulose. The genetic material is not enclosed in a nucleus. It's a single DNA loop, and there may be one or more smaller rings of DNA called plasmids too. And we've also reminded ourselves that those important ribosomes are in all cells, and they're the site of protein synthesis. But prokaryotic ribosomes are smaller than those in eukaryotic cells. Okay, time for a GCSE exam question. So by all means, you can use the PowerPoint to print this off and write on, or you can just have a go on some paper at the side if you can read all the questions. And pause, and then we'll go through the answers. Okay, so going through these answers. So the cell contains a nucleus. What's the function of the nucleus? Controls the activities of the cell. And you're also allowed to say contains the genetic information, or contains the genes, or DNA, or chromosomes. Any of those would be fine. Name one type of cell that does not contain a nucleus. Now the answer here is interesting. Most of you, from what we've recapped, will just say a prokaryotic or bacterial cell. But you wouldn't be wrong if you talked about xylem cells or red blood cells also not containing nuclei. But that's something we'll recap a little bit later. Simple diagram labelling two parts of the cell. So if you've recalled the diagram we did earlier, you'll get your 2 marks. They wanted a bit of a similar shape to the one in the exam question. So I might be better if my nucleus was a bit smaller and a bit higher up. Name one structure found in a plant cell but not found in an animal cell. So those were the ones that we've outlined as being different: a cellulose cell wall, chloroplast, and that permanent vacuole. So we'd have got 100% hopefully on that one following our recap. Quick quiz to check. Okay, do this one in real time. So just stay with me. I read the answer. You think about what you think the answer is as quick as you can. So, which organelle is the site of protein synthesis in all cells? The ribosomes. I'm hoping pops straight into your memory. Which organelle is the site of photosynthesis and so found in plant and algal cells? The chloroplast. Number 3. In what type of cell are the smallest ribosomes found? Quick as a flash, I'm hoping. Prokaryotic cells. So the prokaryotic ribosome is a 70S ribosome, okay, which is smaller than eukaryotic ribosomes, which we refer to as 80S. Number 4. In what cell type is the DNA circular and free in the cytoplasm? Straight away, I'm hoping prokaryotic cell or bacterial cell pops into your brain. Question number 5. Name the part of the cell which controls the movement of molecules into and out of the cell. No matter which cell type, it's always the cell membrane. So that's the same role in all cells. Question number 6. Name the part of a plant cell which gives the plant cell its mechanical strength. So that helps it to resist osmotic pressure when water moves in. The cell wall. Question number 7. Now, did you pick this one up? Name an animal cell which does not have a nucleus. I'm hoping red blood cell pops straight into your mind after that recap with the exam question. You remember they're specialised so that they can contain more haemoglobin, and they've got that bioconcave shape to increase their surface area to volume ratio for the diffusion of oxygen across their surface. By having no nucleus, they can have more haemoglobin and so carry more oxygen. And number 8, can you name a plant cell that doesn't have chloroplast? This was always a very common question at GCSE. Once we've taught you the general cell structure, there are a lot of plant cells then that we then look on at in our specialised section that don't have chloroplast. I'm hoping you got one. So you could have had the xylem or the her root cell, or even the phloem, actually, you can have as well. Okay, so thinking about specialized cells, can you name all of these? Okay, so you can pause and write them as quickly as possible. Off you go. Okay, so we there or thereabouts? If not, just pause me and start me again in a minute. Okay, so the first one is our palisade cells. So they're the leaf cells, specialised by having many chloroplasts. Okay, those chloroplasts are pushed to the side of the cell so that they are more likely to be struck by light. Okay, so many of them, because the specialisation of this cell type is it's a leaf cell for photosynthesis. Next one, skin cell. Then we've got our nervous cell or neuron. Then we've got our red blood cell, as we talked about, specialised in having no nucleus. It can have more hemoglobin present in the cell to increase the amount of oxygen it can carry. Then we've got a sperm cell, her root cell again, another plant cell, but a specialised root cell. Then another plant cell here, we've got the guard cell around the stomata, which you would have talked about when you did transpiration. Then we've got some muscle cells, and we'll look at those 3 different types of muscle that we need to know about. And finally, our white blood cell, which are the immune cells that you've talked about in your immune response topic. So those are your B and T lymphocytes and your phagocytes as well. Right, so thinking about those specialised cells, these are the ones listed in the specification at GCSE. So once you learn the generic animal cell structure, then your teachers will have gone on to talk to you about how those cells are specialised in multicellular eukaryotic organisms, particularly humans, because obviously we are a human and we like to know about our body systems and our body tissues, and so our cell types that make them up. So things that you might know about the sperm. So specializations, there is within its neck, or just downstream, its middle piece here, just underneath the neck, an area packed out with mitochondria. Okay, and that's because the sperm requires lots and lots of ATP for the movement of its tail. So this is like a whip-like tail that allows it to swim. So that mitochondria allows lots of cellular respiration to produce lots of ATP. So that's an energy storage molecule. So that's energy producing. We can't say producing energy even at GCSE. So producing ATP. Energy in the form of ATP for the movement of the tail. You'll know about the nucleus as well, only containing half the chromosome number, one of each homologous pair, produced during meiosis. And you'll know about, in the head, a little packet of digestive enzymes. That's called the acrosome. And that's what allows the head of the sperm to penetrate the egg during fertilization. Okay, so then you'll have looked at neurons. You would have talked about the neuron being a long, thin cell. That's a good specialisation because it's carrying the nerve impulse from within the nervous system along from the cell body to the axon endings. Okay, and you've talked about sensory neurons, relay neurons and motor neurons in your studies of the nervous system. As well as being long cells, you will find lots and lots of mitochondria down the axon of the cell because conducting nerve impulses is very, it costs a lot of energy as well, so you need to produce a lot of ATP. And then you will have looked at different muscles in your studies. You will have talked about skeletal muscle. And they're the types of muscle that are attached to your skeleton. We'll look at them in lots of detail at A level. the cardiac muscle, which we'll link back to in our later video on the heart, and the smooth muscle. Now that's the muscle you find in your blood vessels and also in your gut. Okay, so all of these are different types of specialized animal cells. Then you will have also looked and met the detailed sort of functions of each of these cells, particularly when we were talking about the movement of water, mineral ions and sugars through the plant. So we've got the hair root cell, specialised by having an elongation to its cell structure. That's got lots of mitochondria too. This elongation gave it a large surface area to volume ratio. That was to allow the diffusion of many mineral ions and water by osmosis into the cell. Then the xylem and the phloem are funny old cells in the plant because really they are vessels, and particularly our xylem has none of the cell content that you would expect in a normal plant cell. It's got a really thickened, lignified cell wall, and when it's mature it has no subcellular structures at all. So it's a dead cell that just becomes a tube through which water moves in the plant. Having a look at the phloem, that's not a dead cell, it's a living cell, but it does have reduced numbers of things like mitochondria. And it has these companion cells next door to them that take over some of the roles that that cell can't do because it's got a reduced number of subcellular structures. But they are also long and thin and form tubes, not continuous tubes. So these side walls are a bit like sieves. So we call them sieve tubules or sieve elements. Okay, so those are cells you've met and you will meet again at A level in our study of specialised cells in more detail. Okay, so moving on to some practical work. So you should have done some microscope work at school. We'll do loads more at college. There's 4 questions for you here. Thinking back to your microscope work, this is the kind of microscope that you'll have used up in the top corner. Can you give the parts of both animal and plant cells that you cannot see with this kind of microscope, which is called a light microscope? Number 2, using the terms resolution and magnification. Can you explain why you cannot see these structures with this kind of microscope, this light microscope? Number 3, can you give the name of a microscope that you could use to view these structures? And then finally, number 4, what's wrong with my microscope diagram? Okay, you can pause there and have a go at them all if you haven't finished. If you've finished, just review the answers with me. So we're looking for structures that are small, and so the light microscope will not allow you to see them. So things like mitochondria and chloroplasts, unless you've got a very good microscope. There are some microscopes with shorter wavelengths that you can get now. Generally the ones that we have in our laboratories in schools and colleges are not. So they're just a bog standard light microscope, and things like mitochondria and chloroplasts you cannot see. And the reason for that is the light microscope has a low resolution, and so it's limited to a low magnification. So you cannot view objects this small using that light microscope. Only things like the nucleus is big enough. Obviously you can see the boundary of the cell because the whole cell is big enough, and that's so you can see the boundary, which is your cell membrane in your animal cell, the boundary which is the cell wall in a plant cell. So what kind of microscope would you use if you wanted to visualize, wanted to see mitochondria and chloroplasts? Well, an electron microscope. An electron microscope uses a beam of electrons instead of light. And a beam of electrons has a much shorter wavelength than light, and so it gives it a greater resolution and allows you to magnify to much greater magnifications. So what's wrong with this diagram? Lots. There was no scale or magnification on the diagram. I shaded the nucleus, which is a big no-no, isn't it? We don't shade. The size of the pencil I used to draw that diagram, my word, those are thick lines, so that's no good. It's a bit sketchy. So you can see here, it's a little bit sketchy. There's a bit of overlap there. So not brilliant. And there are no labels at all, so that's no good. Okay, so no labels. So a bit of a reminder then, our electron microscope, that has that much higher magnification and resolving power than the light microscope. And that was because the beam of electrons that's used by this microscope to create images of the insides of cells, it has a much shorter wavelength. This means you can study inside of cells in much finer detail. And this has enabled biologists to see and understand many more of these subcellular structures. Now at A level, we call the subcellular structures organelles. And the organelles that we haven't met at GCSE yet are the ones that were discovered using the electron microscope. So there's a whole host yet for you to learn about, which if you do some of the extension materials to this topic, you can find out about. Things like the Golgi apparatus and the rough endoplasmic reticulum. Okay, so sometimes people struggle a little bit to understand resolving power, okay? So when we say resolution, if you think about, I suppose now when you are looking at resolution, you might think about your phone or something like that. If the image that's produced has poor resolving power, low resolution, it means two images that are quite close together appear as one image. Whereas with the electron microscope, two tiny objects really close together still are visualized in the electron micrograph, the picture produced by the electron microscope, as two separate objects. For the light microscope, when you start to try to look at smaller objects to increase your magnification, it goes blurry like this because it's not got the resolving power. So our wonderful electron microscope can show us those finer details because it has got that high resolution, that high resolving power. So this has hit in an exam question last year. Okay, so students were asked the two advantages of using an electron microscope instead of a light microscope. So you can pause and have a go. Okay, so have you got the idea that for one, we can have, that gives us a higher magnification, and for two, a higher resolution. Yeah, so we can see more detail inside the cell, we can see smaller objects, we can see smaller subcellular structures. Any of those ways of describing higher resolution. Okay. So the next skill I want to recap is magnification calculations. This is a skill you are going to use in your cells topic and other topics as well in your A-level biology. Okay, so a bit of a recap over there on the side. So if you've got the image size, this is the thing that you can measure with a ruler on the image in front of you, and you've got the magnification, you can divide that image size by the magnification, and that will give you the real size, the actual size of the object. Often when we do that though, our answer will be in an inappropriate unit. Okay, so if we're looking at an image of a mitochondria and we measure that image in millimeters and then we divide by the magnification, our answer will still be in millimeters. So at some point we need to think about changing the units to a more appropriate unit you would have for something small like a mitochondria, and that would be a micrometer. Okay, now we need to be able to rearrange that formula when we want to calculate different things. So sometimes you'll be given the image size and the actual size, and they'll want the magnification. Sometimes they will give you the actual size and the magnification and want the image size. That one's the least frequently asked. Okay, so we're going to practice that skill. So you want to take that down, you can. I think another thing to remind yourself of is the conversion from micrometers to millimeters and millimeters to micrometers. Okay, so in each millimeter there are 1,000 micrometers. So if you're in millimeters and you want to get to micrometers, you times by 1,000. If you want to get from micrometers to millimeters, you divide by 1,000. Okay, so you can either print this out from the PowerPoint or you can just do it here from the video, but could you have a go at these calculations? Okay, so in terms of our description here, I've used real size instead of actual. Okay, this is my actual size. So in the first one, the actual size is 20 micrometers. The magnification is times 5,000. What is the image size? So this is just practicing and recapping what you have done at school. Okay, so I'm going to go through the answers to that one. So if you're not quite ready, pause the video. Okay, so the first one, we take our real size, okay, because we've rearranged this, we've got our real size, we've got our magnification, so we times them together to give us our image size. Okay, so when I did that, it gave me a really big figure. Okay, so the image size in micrometers, that wouldn't be appropriate, so I've converted it by dividing by 1,000, so my answer's in millimeters. The next one, the real size was 7 micrometers, the image size 6 millimeters. What's the magnification? Make sure you keep, this is a reminder here, one thing students forget to do is before applying the formula here, if you've got your image size divided by your real size, you need to make sure they're in the same units when you do that division. So convert one before. Okay, so doesn't matter which one you convert, just make sure they're in the same units. I tend to go for this one where I get my image size and times it by 1,000. That's the one I put on top and then divide it by my real size, and that gives me the answer times 857. Doesn't matter if you did it the other way around and converted the bottom to millimeters by dividing by 1,000, as long as they're in the same units when you do the division. Okay, so on the next one, we've got image size, magnification. What's the real size? So the image divided by the magnification gave me the real size in millimeters, not appropriate, so times it by 1,000. Now I've got it in micrometers. Now, this one might have tricked you unless you did a bit of Google search. Okay, so I'm not sure at GCSE you get introduced to this unit. It is a nanometer. Okay, so there are 1,000 nanometers in a micrometer. Whew. So there's 3 different ways you could have done this one. Okay, so probably this would be the way I would go for. So if it's 1 millimeter, I times it by 1,000 to get it into micrometers, and then times it by 1,000 again to make it into nanometers, and then I can divide it by my real size, actual size, which is 70 nanometers, and I get my magnification. Okay, going back to the more simple ones now. Real size 17 micrometers, image size 2 millimeters. What's the magnification? So I've got my image size, my actual size in the same units, doesn't matter which way, and I get my magnification. Okay, so I'm hoping that recap just reminded you of that skill. It's a really important skill to take through to your A-level biology. Here's an example of an exam question you can have a go at now. This is from the GCSE last year. If you want to pause it and have a go. Okay, so once you have a look, this is your answer here. So they've told us that from X to Y was 0.06 millimeters. Okay, so that's my image size. Not image size, that's my real size. When measuring, that's the measurement that they got. So from here to here, sorry, I should have given you that before. Don't worry, I have on the next one. So from there to there, they would have measured. Now, it's really important here, I would say, to make sure you measure in millimeters. So to do this one correctly, you would have measured from X to Y at 24 millimeters. That would have given you your image size. You would have times that by 1,000 to convert it into micrometers. Oh no, you wouldn't. You would have kept it the same, sorry, because they've given you this in millimeters, the real length. And so you would have done 24 divided by 0.06. So that would have been your calculation, and you would have got times 400. If you'd have done that, they did give you credit on the exam, but you'd have been out by a factor of 10. So for A-level, please remember to measure in millimeters. Okay, so have a look then at these examples that I've put together for you. So we're measuring the distance that the oxygen would have to move from the alveoli to the red blood cell by measuring this bar A to B, and it is 21 millimeters from here to here, and that represents a distance of 1.5 micrometers. Okay, so for this one, what would we do? We've measured at 21 millimeters, always measuring in millimeters. That's our image size, that goes on top. This is our real or actual size, that goes on the bottom. We want them in the same units, so I would times the top one by 1,000 to convert it to micrometers, but you could do the opposite to the bottom. Do my division, and that's what you should get, times 14,000. Okay, on the next one, this is a bit different. Sometimes you're given a scale bar. Okay, so in this example, the scale bar was just on the diagram, and the scale bar showed you 5 micrometers. So in other words, this distance on this diagram here is 5 micrometers. Now this one's taken from an A-level script, and you can see that we're using the same concept that you learnt at GCSE, but we're applying it now to the scale bar. Now don't worry if this sounds a bit weird, because we will do lots of practice of this, but we're measuring it. We get our our image size here. The real size is the 5 micrometers on the scale bar. So now I can do my 24 millimeters, times it by 1,000 to make it into micrometers, divide it by 5, and that gives me the magnification of the image. And that's without measuring anything on the image, I'm just using the scale bar. Okay, so what have you got to look forward to studying cells at A-level? Well, as you can see from our recap, you already know loads of things about the differences between subcellular structures within plants, animals, and prokaryotic cells, which are bacteria. But when you come to John Rigby and you study A-level biology with us, we're going to take you to much greater depth. You're going to get introduced to new organelles, so new subcellular structures and their functions within the cells. You're going to keep going with these magnification calculations as I've just shown you, and they're going to go from, I suppose we're in that GCSE example from 2019, you were just doing a measurement and then using the formula you were given in the exam, to things like this at A level where you're given more of a problem to sort out, or even applying it to a scale bar. For your microscopes, we will look at electron microscopes and light microscopes again, and we'll compare them in more detail. You'll be asked to apply your knowledge a lot more when we're studying our A-level exam, so we might even be shown cell types we haven't met before, and from those structures be able to suggest how their adaptations might allow them to do particular functions. Those for known, like the ones that we've started to look at GCSE, the nervous cells, the neurons, muscle cells. And then we're going to start to link the roles of the organelles together. So when we think about, currently you know about the mitochondria producing ATP, providing the energy to the cell's activities. We know that the proteins are produced at the ribosome, but at A level we'll start to think about how those two jobs are linked together and the other organelles involved in, for example, protein trafficking. And finally, this is an incredibly important topic because it underpins all the specialised cells we'll meet at A level, from the sieve cell in the xylem to the motor neuron again. Okay, so not leaving you quite there. There's an example there of an A-level challenge question if you want to have a go at that. And an extra GCSE exam question. And you're welcome to send your answer to those questions to me at my email, or you can contact me if you've any questions at all, or if you want any additional support resources in your transition period. So that's sort of the finished video for cells. I do hope that that's useful to you. We'd love some feedback if you want to send that to me on my email as well. And we hope to see you very soon at John Rigby. So that's bye for now.